All right, welcome to our genetics and heredity review. This is part 2 of a 5-part series that reviews everything that we've covered this year in biology. So, genetics is just the branch of biology that studies heredity, which is the passing of characteristics from parents to offspring, and we call these characteristics traits. So we're going to start our genetics portion of the review, and then we'll sort of transition into heredity. So I want to start by reminding you that DNA is one of our complex biomolecules, one of our macromolecules. We learned it as a nucleic acid. So if you missed the first review, the cells review, you're going to want to go back and listen to that review because we talked about the 4 macromolecules, and one of those was nucleic acids, which includes DNA and RNA. So these nucleic acids are made of monomers called nucleotides, and you can see an image of a nucleotide here. You're going to have to memorize the 3 parts of a nucleotide, so just make sure that you remember that a nucleotide contains a phosphate, a sugar, and the phosphate and sugar make up the backbone of the DNA or RNA molecule. Now, this sugar is going to differ depending on whether we're talking about DNA or RNA. So in DNA, it's called deoxyribose, but in RNA, it's going to be called ribose. And then we have a nitrogenous base. And that nitrogenous base, there's 4 options in a DNA molecule. So we can have adenine, thymine, cytosine, or guanine. And then in an RNA molecule, we can have adenine, uracil, cytosine, and guanine. So this is just the 4 nitrogenous bases that I just mentioned listed here. And then just remember that the structure of a DNA molecule is double-stranded. We say it's a double helix. And we have base pairing rules that are going to be important throughout the review. We're going to come back to these. Remember, adenine always pairs with thymine. So A always pairs with T. You can remember that apple in a tree. And then C always pairs with G. So cytosine always pairs with guanine. And you can remember that because cars can be found in a garage. So the big overall question that we can answer through what we know about genetics and heredity is how can organisms be so different if their genetic material is the same molecule? So if we only have 4 options for nitrogenous bases, then how is it that we're so genetically different from other organisms? And the answer to that has to do with the order of the nucleotides. So the order of the nucleotides vary from organism to organism, and this creates what's called genetic variability. So DNA has the unique ability to make an exact copy of itself, and we call this DNA replication. We spent some time on this year, so this is kind of going way back. You may not remember everything about DNA replication, so I'm going to try to just hit the high points here. So during DNA replication, an enzyme called helicase unzips the hydrogen bonds that hold the nitrogenous bases together. This is a weak bond, so it's easily unzipped, and the DNA strand unwinds, and this allows the free nucleotides to bond to the single strands that are open. And DNA polymerase is going to ensure that the nucleotides are matched using those base pairing rules, so A's and T's and C's and G's. Now, this process is going to continue until the whole entire molecule has been replicated. Each new strand is formed. We call this a complementary strand. So what we end up with at the end of DNA replication is 2 strands of identical DNA. And each strand has one side that contains the original parent strand, and then the other side is made up of a brand new strand. So we call a replicated DNA molecule semi-conservative for this very reason. So this replicated DNA will be passed to new cells through the process of mitosis or to new generations through meiosis. Just a reminder, in eukaryotic cells, DNA is found in the nucleus, and it's coiled into chromosomes. I have a picture of that in just a second. I'll show you. And we can have prokaryotes that also contain DNA, but remember, since prokaryotes do not have a nucleus, their DNA is going to either be attached to the cell membrane or free-floating in the cytoplasm. Now, I don't want to forget about RNA, so you need to be able to compare and contrast DNA and RNA, and while there are some similarities, there are also some differences. So let's go through some of the important similarities and differences. So just a second ago, I mentioned that DNA is made up of there's four options for nitrogenous bases: adenine, thymine, guanine, and cytosine. You're going to notice that three of those nitrogenous bases are also found in RNA, so adenine, guanine, and cytosine. But in RNA, we're not going to have thymine. Thymine is going to be replaced with uracil. Now, also, you probably remember from the slide earlier that DNA is a double-stranded molecule. RNA is just single-stranded. And the sugar is different. I didn't put that on the slide, but there's a difference in the sugar. So earlier I mentioned that DNA is made of deoxyribose. RNA is made of ribose. Now, the base pairing rules are going to be a little different from RNA since we don't have the thymine. So with DNA, A paired with T, but since T is going to be replaced with uracil, A is going to pair with U when we're talking about RNA. And then C pairs with G, just like with DNA. All right, so I might give you one side of a strand of DNA, and I might ask for the complementary side or complementary strand. And so since I'm asking you to find essentially the other side of the DNA molecule, you're going to use base pairing rules for DNA. So A pairs with T, and then C pairs with G. G pairs with C. A pairs with T. Since we have thymine on the left, we're going to have adenine on the right. And since we have cytosine on the left, we'll have guanine on the right. And then guanine on the left, which means cytosine will be on the right. So just make sure that you know you're going to use DNA base pairing rules if I give you one strand of DNA and I ask for the complementary strand. All right, moving through, hopefully you remember talking about protein synthesis this year. So protein synthesis is just the process in which cells make proteins. And this happens in two stages. So if you'll look at the screen, you'll see that DNA has to be transcribed into RNA, and then that RNA has to be translated into a protein. So to make proteins, this process has to happen. We call this the central dogma of biology, but it's protein synthesis in a nutshell. So from DNA to RNA, we call this transcription. And then when we take RNA and ultimately create a protein from that, we call that translation. You'll need to remember that transcription happens first and translation happens second. And an easy way to remember that is these words are in alphabetical order. So if you're trying to recall which one of these happens first, transcription, if you were putting these words in alphabetical order, transcription would come first. So that might be a good way to help you remember. Transcription comes before translation. Excuse me. All right, so let's take a look at the two steps separately so that you remember. On your end of course assessment, you're probably going to see some diagrams where you have to identify, hey, this is transcription or this is translation. So just remember that transcription happens in the nucleus. So we're going to see the DNA first. The DNA can't leave the nucleus. So what's going to have to happen is mRNA is going to have to be created, and DNA is going to be used as a template to make that mRNA. So the DNA sequence or the DNA strand unwinds, and the mRNA starts to be created from the DNA template. And then that DNA or that mRNA is going to leave the nucleus. It's going to carry the genetic information in the form of mRNA, and it's going to carry that information to the ribosome. So at this point, we have RNA, and we're moving into translation, which is the second step of protein synthesis. And ultimately, we're going to turn that mRNA into a protein. So once at the ribosome, the process of translation just kickstarts. So we have our mRNA strand here, and it's going to be read by a tRNA molecule. Sorry, I was trying to remember what picture I had coming up. So we have a tRNA molecule that's going to come in, and it's going to read the codons. So remember, codons are a set of 3 bases that code for amino acids. So this tRNA will read those 3-letter codes and it's going to drop off the appropriate amino acids. And then these amino acids will link together, and once they link together, we'll have a protein. Let me go back and let you look at that. All right, so let's say I ask you to transcribe a DNA molecule. So transcribe, we're thinking transcription, the first step of protein synthesis. So this is where we're going to turn DNA into mRNA. So we're going to use the DNA as a template to create our strand of mRNA. Now, since we're trying to create RNA, we need to use RNA base pairing rules. So we won't see thymine when we're creating an mRNA molecule. We're going to replace that with uracil. So T pairs with A, and then A is going to pair with U. Cytosine pairs with guanine. Guanine pairs with cytosine. C pairs with G, and A pairs with U. I might also ask you to translate the following mRNA molecule into an amino acid sequence. So if I ask you to translate something, that's the second step of protein synthesis. That's translation. So what we're going to do is we're going to break our mRNA into codons, so chunks of 3. We're going to start here with AUG, and then you're going to have to, when you're trying to find the amino acid sequence, you're going to have to use the codon chart. So here I have a codon wheel. Remember, you read from the inside and work your way out, and that will uncover the correct amino acid. So I have AUG. So if I'm looking at my codon wheel, A, and then U, and then G, this is methionine. And then we have CGU, which is arginine. And then we have UAC, which is tyrosine. Okay, so our 3 amino acids are met, arg, and tyr. All right, so it is important to understand there's a lot of vocabulary associated with genetics and heredity, so cells, chromosomes, DNA, genes, alleles, and it's important to wrap your head around all that vocabulary that we talked about this year. So this picture does a great job of trying to just unlock all the different vocabulary terms that can sometimes be confusing. So starting with the cell, inside the cell you have the nucleus, and in the nucleus you have chromosomes. And then if you unwind those chromosomes, we have DNA. And then if we focus on a segment of DNA, these are known as genes. And we learned this year in heredity that genes code for traits. So this could be eye color, hair color, height, fur patterns. Now, genes are located on chromosomes, and they can exist in alternative forms, and we have a special vocabulary term for that. We call that alleles. So here you can see my chromosomes. You can see that I have a big B and a little b here. So on my chromosomes, I have genes. I've identified those genes and labeled those genes. And when we have different versions of genes, we call these alleles. So this capital B might represent brown hair, and this lowercase b might represent blonde hair. So we're looking at the same gene. This is coding for the same trait, but because we have different versions of that gene, we're just going to call this alleles. So alleles are found on different copies of chromosomes, and one of these genes or alleles is going to come from mom, one comes from dad. All right, we also talked about genotype and phenotype this year. So just to recall those vocabulary terms, genotype is just a list of alleles for a particular trait in an organism. So we use letters to represent genotype. So I might have big B, or little a, or one capital and one lowercase letter. And then we talk about phenotype. We're talking about the physical appearance of an organism. So how these alleles influence the function of that particular gene. So some examples of phenotype could include blue eyes, spotted feathers, or even blood type like AB blood type. When I have 2 capital letters in genetics or heredity, we use capital letters to represent dominant alleles, and then we use lowercase letters to represent recessive alleles. So if I have 2 capital letters that I'm using as my genotype, then we call this homozygous dominant. If I have two lowercase letters that I'm using for my genotype, we call this homozygous recessive. And then if I have one capital letter, one lowercase letter, they're different. So we call this heterozygous. Now we use genetic crosses in in heredity. We use genetic crosses to just determine the probability that a certain trait will show up. So genetic crosses that involve one trait are called monohybrid crosses. These are a more simple form. And then we can get really complex. So this year we also talked about dihybrid crosses where we cross two traits. These are a little more complex. Now, the outcomes of genetic crosses can be predicted by using laws of probability, and Punnett squares just give us those possible results. So let's take a look at an example, just in case you may have forgotten or it's been a while since we've done Punnett square. So let's just do a simple Punnett square just to remember how it works. So in rabbits, black fur is dominant over brown fur. And if one parent rabbit is heterozygous, and the other parent is homozygous brown, so that's going to be little b, what's the probability of producing an offspring with brown fur? So I'm going to cross these individuals, and so this is the likelihood that we'll have rabbits with black fur versus rabbits with brown fur. So I've got some specific questions I'm going to go through with you. The first question is what percent of the offspring would be black? So it does tell us in the problem that black fur is dominant over brown fur. So if I see a capital letter in these Mendelian genetics problems, the dominant trait's going to show up. So I have two squares where I'm going to have black fur and two squares where I'm going to have brown fur. And so the percent chance that the offspring is going to be black is going to be 50%. So 2 out of 4. We also have 2 out of 4% chance that the offspring will be brown. So that is 50%. And then I asked for the genotypic ratio. So remember, genotypes are letters. So for every 2 individuals that are big B, little b, 2 individuals will be little b. So a 2-to-2 ratio will simplify to a 1-to-1. And then the same thing for phenotypic ratio. For every 2 individuals that have black fur, 2 are going to have brown fur. So a 2-to-2 ratio would simplify to 1-to-1. All right, we talked a lot this year about Gregor Mendel and his work with heredity. And he has 3 laws. So his work can be summarized in 3 laws. We're going to quickly go through these. So the law of dominance just states that the dominant allele will prevent the recessive allele from being expressed if they're paired together. And the recessive allele will only appear if it's paired with another recessive allele in the offspring. So here in our example, we have a cross between 2 heterozygous individuals where the purple flower color is the dominant trait. So therefore, we're only going to see the white recessive trait when 2 recessive alleles are expressed. So we do see that here in the bottom right-hand corner. So we're going to get the white flower. All right, so the law of segregation is Mendel's second law. It's also known as the law of separation, and it just states that gene pairs separate when gametes are formed. So you can see here in this picture, through meiosis, gametes, 4 daughter cells are going to be created. We call these gametes or sex cells, and you'll notice that each sex cell only has 1 allele of each pair, and that's known as the law of segregation. So these chromosomes just kind of segregate out. There's also the law of independent assortment, and this just states that different pairs of genes separate independently of each other when gametes are formed. So during the process of meiosis, there's so many different combinations, the way that they separate themselves out and assort themselves. There's so many different allele combinations. This creates genetic diversity, and Mendel recognized that, and he recognized that and coined this law. So law of independent assortment. All right, so there are a couple of instances where Mendelian genetics just is not a factor. So there's some exceptions to the rules. One of those exceptions that you need to make sure that you know is codominance. So in codominance, both alleles are going to show up fully. So here's a good example. So here we don't have dominant and recessive, both alleles are going to show up. So I might have a white cow crossed with a red cow, and I might get what's called a roan cow, which both colors are going to be fully and separately expressed. In class this year, I told you could remember this, but by thinking about a co-pilot. So co-pilots, when you fly, you have more than one pilot, just in case something happens and you need a backup, right? So co-pilot, both pilots are showing up to work that day. In codominance, both alleles are going to show or be expressed. In incomplete dominance, we're going to see a mixture of alleles. So again, you don't have dominant and recessive, you're going to get a mix of alleles. So in incomplete dominance, I might have a situation where I have a white flower crossed with a red flower, and the colors are going to mix. And so I might get offspring that have a mix of the two variations, and I might have pink flowers. All right, we know that traits are passed from parent to offspring, and offspring are created through sexual reproduction, which is the union of an egg and a sperm. And in biology, we refer to eggs and sperm as sex cells, which are also called gametes. And in our last sales review session, we talked about the process of meiosis, but just remember, meiosis is the process where gametes are made, and you can remember that. Some people call meiosis, which is fine. So if you pronounce meiosis, it would be easy to remember, because meiosis, think about me. This is how I am made. It is very, very important that you know the process of meiosis. So it starts with a single diploid cell. This is our parent cell. And we're going to go through 2 rounds of cell division. So remember, with mitosis, we only have 1 round of cell division. But in meiosis, we're going to go through PMAT twice. So prophase, metaphase, anaphase, telophase, and then again, prophase, metaphase, anaphase, telophase. And what this does is it takes that one parent cell and it turns it into 4 daughter cells. And these daughter cells are haploid cells, so they're going to have half the number of chromosomes as the original parent cell. Now, fertilization is going to restore that original number of chromosomes. So while our gametes contain half the number of chromosomes as the parent's body cells, that number will be restored at the union of the egg and the sperm. So during reproduction, when the egg and the sperm come together, or fertilization rather, when the egg and the sperm come together, that union is known as a zygote, and that restores the full number of chromosomes. So egg cells are haploid, sperm cells are haploid. So all gametes are haploid. But when they unionize and the egg is fertilized, that union is a diploid cell. All right, so we can use that to answer some questions. So this says a goat's somatic cells contain 60 chromosomes. So how many chromosomes do each of the goat's gametes have? So remember, gametes are going to have half the number of chromosomes as somatic cells in the organism. So you're just going to take 60 and divide it by half, and your answer would be 30. Humans have 46 chromosomes in each of their skin cells. So how many chromosomes can be found in a human gamete? Again, gametes are going to have half the number of chromosomes as body cells. So half of 46 would be 23. All right, we have lots of sources of genetic variation. So one of the terms that kept coming up this year is genetic variation, and how individuals can be genetically different from one another. And so there's different sources of genetic variation. We talked about these sort of sprinkled throughout the year. So this is the first time we've seen them collectively all together. So independent assortment is one source of genetic variation. Again, this is the way that chromosome pairs line up at the equatorial plate during meiosis. And this influences how they're distributed into the gametes. And so you can look at this picture and see there's a lot of different allele combinations, and that's because of independent assortment. We also have crossing over that happens during meiosis, specifically in prophase 1. So this is where chromosomes physically overlap and they exchange chromosomal material, and this just is another way to create variation in individuals. We can also have mutations, and if you'll remember, mutations are just changes in the nucleotide sequence of a DNA molecule. Gene mutations can be the result of exposure to agents like UV light or radiation, also free radicals or chemicals that are found in the environment, substances in tobacco products, and even viruses. And all of these agents are called mutagens. Gene mutations can also be the result of an error during DNA replication. So earlier we talked about DNA replication. If that process doesn't happen seamlessly, that could create a gene mutation. There are different gene mutations that we talked about this year, 3 specific that you need to remember. So we can have substitution, insertion, and deletion. So just to review, substitution occurs when one nucleotide base is replaced by another. So here in this picture, we see a normal, the way it's supposed to normally work. And this is a normal sequence. And then we have a mutated sequence in the second picture. This is an example of substitution. So here we're supposed to have cytosine, but that cytosine has been replaced with thymine. And so that replacement causes the wrong amino acid to be coded for. And so that creates a mutation. A great example of a substitution mutation is sickle cell anemia. We talked about that one this year. I just wanted to remind you that that was an example of a substitution mutation. You can also have an insertion mutation. This is going to occur when one nucleotide base is added to the sequence. And what this is going to do is it's going to cause a frame shift. So everything's going to be shifted down. So you can see here we have inserted an extra cytosine here in our nucleotide sequence. And when we add in that extra nucleotide, it shifts everything down. And so what that did in our sequence is it coded for the wrong amino acid, which is what creates a mutation. You can also have deletion. This is going to occur when one nucleotide base is removed from the sequence. This also generates a frame shift. So here we were supposed to have an adenine, but it's been completely removed in the sequence. And so you can see here, because everything shifted down, it created a difference in the amino acids that were supposed to be coded for, which could create a huge problem. You can also have chromosomal mutations, which are going to be changes in the number or the structure of a chromosome. And we can use a karyotype chart to recognize some of these chromosomal mutations. So we're supposed to, in the karyotype, humans have 23 chromosomes, and we're supposed to have homologous pairs here. But if you notice on chromosome number 21, there are too many chromosomes. This is known as trisomy 21, which is Down syndrome. You can also have something called non-disjunction. So what's going to happen here is the chromosomes are going to fail to separate properly during cell division. This can happen during mitosis or meiosis. So what happens here is a lot of times this will happen in the transition from metaphase to anaphase. So the chromosomes are supposed to be pulled apart. Occasionally, the spindle fibers won't attach properly, and this creates non-disjunction. So you'll wind up with daughter cells that have too few or too many chromosomes in them, and so that's just an example of non-disjunction. Now, we have learned so much about DNA in the past 20, 30, 40 years. And so this information, the things that we know about DNA technology and the research that's been done, is being used in a lot of good ways. And so we've talked about some of these good things that DNA has allowed us to do, or what we know about DNA has allowed us to do. We have advancements in medicine and forensics and agriculture. So we're going to go through some examples that we discussed this year. Certain genetic diseases can be cured by reinserting a corrected gene into a patient to replace a damaged gene. We call this gene therapy. In forensics, we can find crime scene DNA and match it to potential suspects to solve crimes. In agriculture, plant biologists and even farmers have been able to work together to use DNA technology to produce GMOs, or genetically modified plants. And this does a lot of different things for the farmer, but we can create plants now that have increased disease resistance or herbicide resistance, or even increased nutritional value. Researchers use something called recombinant DNA technology to analyze genetic changes. So basically, they cut and splice and insert modified DNA molecules from different species into these things called bacterial plasmids. And then the cells copy the foreign DNA right along with their own DNA. And so that's a form of genetic engineering that then allows us to do some of the things that we've talked about. A great example of this is human insulin. So insulin can help many people that have type 1 and type 2 diabetes, because insulin can be mass produced, and that's all due to genetic engineering. In agriculture, there's a lot of different ways that farmers are using genetic engineering. Here's an example. So it says the buds of cotton plants were vulnerable to worm attacks. And so through genetic modification, we can create cotton plants that now resist these worms, and this results in increased cotton production. So it benefits the farmer. We have even made genetically modified bacteria that can help the environment. When there's an oil spill, it can eat up the oil. So that's awesome that we're using DNA technology and genetic engineering to solve real world problems. However, with all the benefits comes some risk. And so it's important to weigh out both the benefits and the risk of using GMOs and genetic engineering. So some of the benefits, we do get desired traits. We can mass produce a lot of different things in both agriculture and medicine. We can use fewer pesticides in agriculture because of genetic engineering. It's typically cheaper, well, in some ways it's cheaper, because you'll notice on the other side it says expensive. But in some ways it's cheaper. In the medical field, it's more expensive. It's more productive for farmers, and probably the biggest benefit is it is saving lives. But with that comes the question, is it ethical to alter DNA? Some people argue yes, and some people argue no. Since this is a newer technology, we don't really know the long-term effects. So we don't know how it's going to affect us down the road. In some ways, like I mentioned before, it is more expensive. And then one of the big things we talked about this year is when you use genetic engineering, you are decreasing genetic diversity. And this leads to biological resistance, like antibiotic resistance or pesticide resistance. And we talked about those things during our evolution unit this year. All right, that ends our session on genetics and heredity. I will see you in the next session on classification and phylogeny.